Okay, well, hopefully everyone's been able to check back in. Again, welcome back from lunch. My name is Deb Kilday, and I'll be helping to moderate this afternoon's symposium on endocrine disrupting chemicals and their impacts on women's health issues. So we have an action-packed agenda for this afternoon. So we're going to kick off with our first panel session, Hiding in Plain Sight, Water Disinfection Byproducts. And the next panel speakers are Dr. Jody Flaws and Dr. Natasha Menskova. So for our next slide panel speaker, I'd like to introduce Dr. Jody Flaws. Dr. Flaws is a professor in comparative biosciences at the University of Illinois. She earned a bachelor's degree in science and biology from Xavier University and a master in science and biology from Loyola University. She received her Ph.D. in physiology from University of Arizona. Following completion of her Ph.D., Dr. Flaws performed postdoctoral research at Johns Hopkins University and the University of Maryland. Following postdoctoral training, Dr. Flaws accepted an assistant professor position at University of Maryland, where she subsequently was promoted to associate professor. In 2006, Dr. Flaws accepted a position as professor of comparative biosciences at the College of Veterinary Medicine at UC. Her research program has been focused on determining the mechanisms by which environmental chemicals affect the function and ovary and female reproductive system. This research is funded by the National Institute for Health Sciences and has led to publication in over 300 peer-reviewed papers. Dr. Flaws is the recipient of numerous awards, including the Epidemiology and Preventive Medicine Award by the University of Maryland Student Mentoring Award and the Patricia Sokolove Outstanding Mentor Award, the Dr. Gordon and Ms. Helen Kruger Research Excellence Award, and the Pfizer Animal Health Research Excellence Award. The Dr. Flaws has also received the Lifetime Scientific Achievement Award in Reproductive Development Specialty Section for the Society of Toxicology. Dr. Flaws, I would like to turn the floor over to you so that you can begin.
Thank you very much for that wonderful introduction and for the opportunity to present some of our research in this wonderful symposium. I'm assuming everybody can hear me okay, if I don't hear anything elsewise. So what I would like to do today is first give some background on water disinfection byproducts, and in particular, the disinfection byproduct that we're focused on, which is called iodoacetic acid, or IAA. I'll also give some background on female reproduction, particularly the ovary. And then I'll share with you some of our data on the effects of water disinfection byproducts on female reproduction, sharing our hypothesis, specific aims, results, and then leave you with a summary and some conclusions. So first and foremost, water disinfection is a major public health achievement of the last century. Because we're able to disinfect our water, we've dramatically decreased the incidence of waterborne diseases such as cholera, typhoid, and amoebic dysentery. And because we've decreased the incidence of these very serious diseases, we've substantially increased life expectancy. However, when we're disinfecting our water, there's a reaction between disinfectants and organic matter that forms what we call water disinfection byproducts, or DBPs. We now know that there are more than 700 DBPs identified in drinking water, and that these DBPs largely fit into two major classes, the trihalomethanes and haloacetic acids, or HAAs. Haloacetic acids, they share a common structure with acetic acid. And on the right here, I've got the structure of haloacetic acids. These molecules consist of two carbons, including a carboxylic acid and an alpha carbon. And at the bottom, I'm showing three different haloacetic acids: chloroacetic acid, which has a chlorine, bromoacetic acid, which has a bromine, and iodoacetic acid, which has an iodine. So far, we have identified that there are 13 different haloacetic acids in drinking water. In 1998, the US EPA began to regulate 5 of these haloacetic acids, which are listed here. And they were regulated because they'd been shown to be toxic, and a lot of them were shown to be carcinogenic. In 2016, the US EPA added 4 additional haloacetic acids to their list of regulated disinfection water byproducts, and these are listed here. So even though we are regulating some of these haloacetic acid, there are still many that are not regulated because there's not enough information about their toxicity versus their benefit. And one of these unregulated HAAs is iodoacetic acid. And this is the haloacetic acid that I'm going to focus on in my presentation. So one thing about these DBPs in drinking water, including iodoacetic acid, is their levels are highly variable. Depends on the house, the region, the area where somebody lives. And the amount of DBPs that end up in our drinking water depends on the concentration of organic matter in the water, the chemical composition of the source water, the pH, the temperature. Higher temperatures mean more DBPs present in the water, the type of disinfectant being used to disinfect the water, and the concentration of the disinfectant. And this is just showing a water source that's containing DBPs entering into the house. And these DBP levels increase the further that the water source is from the house, because there's more time for disinfection byproducts to enter into the water source. Now we know that humans are primarily exposed to DBP through ingestion of drinking water, beverages made with water, or food that is made using water containing disinfection byproducts. Humans are also exposed through inhalation and dermal absorption, primarily by taking showers or baths, swimming pools, or in steam rooms. Now, previous studies have shown that these DBPs do raise health concerns. They are known in different cell lines and animal models to be cytotoxic, genotoxic, mutagenic, and teratogenic. And previous studies have shown that different haloacetic acids can modulate gene expression and alter stress response to DNA damage, cell cycle regulation, production of reactive oxygen species, and apoptosis. We also know that HAAs pose reproductive health concerns. They can disrupt estrous cyclicity in rodents and suppress estradiol catabolism, which leads to alterations in sex steroid hormone production in female rats. We know in rats that gestational exposure to a mixture of HAAs results in pregnancy loss and eye malformation in rats. And we know from human epidemiologic studies that HAA exposure is associated with low birth weight, small for gestational age, being stillbirth, or having birth defects. However, the effects of HAA on the ovary, one of the predominant reproductive organs in the female, are largely unknown. And so what my lab has been interested in is studying how HAAs, particularly iodoacetic acid, impact the ovary. And so I just want to give a little background on the importance of the ovary. The ovary is the primary female reproductive organ that's required for the production of oocytes or eggs, and it does this through a process called folliculogenesis. And production of oocytes is absolutely required for normal fertility. If you can't produce eggs, you're going to have an animal or a person that is infertile. The second critical function of the ovary is that it's there to synthesize and secrete hormones, and this process is called steroidogenesis. Now, these hormones include estrogens, progesterone, and androgens, which are absolutely required for development of the eggs, monitoring or maintaining estrous cyclicity in non-human primates, or menstrual cyclicity in humans and primates. Maintaining the reproductive tract, fertility, and estrogens coming from the ovary are critically important for non-reproductive functions. You need proper levels of estrogens to have cardiovascular, brain, and bone health. So I want to give a little more background on the functional units in the ovary, which we call ovarian follicles. So this is a schematic of the ovary, and it's showing that that ovary contains many different types of structures. One of the most immature structures is called the primordial follicle, which has an oocyte surrounded by a single layer of somatic cells called granulosa cells. These primordial follicles are too immature to ovulate or produce sex steroid hormones. They have to grow into what we call primary follicles. Those then grow into preantral follicles, where you've got an oocyte, several layers of granulosa cells, and 2 layers of what we call thecal cells. Finally, preantral follicles grow into antral follicles, which are the most mature follicle type. They contain an oocyte, several layers of granulosa cells, a fluid-filled cavity called an antrum, and layers on the outside of cells called thecal cells. Now, these antral follicles are the only follicle type that's mature enough to release an egg during ovulation, and they are the primary producers of sex steroid hormones. So one of the things we wondered about iodoacetic acid, one of these disinfection byproducts, is whether it affects the ability of follicles to grow and to produce sex steroid hormones. So the hypothesis that we tested was that iodoacetic acid exposure inhibits antral follicle growth and steroidogenesis. To test this hypothesis, we use CD1 mice as a model. These are adult mice that are 32 to 42 days of age. We then remove the ovaries from the mice, and we can dissect out the antral follicles and place individual antral follicles in a 96-well culture plate. The beauty of this culture system is that we can treat individual follicles with either a vehicle control, which in this case is just water or media, or different doses of iodoacetic acid. And we can measure follicle growth in this culture system. We can collect the follicles at different times in culture and look at their gene expression or protein levels. And we can collect the media from these culture to measure hormone levels in the supernate. So I'll share with you some of our data from these cultures. So what I'm showing you here is the effect of IAA on follicle growth in vitro. On the y-axis is the percent of follicle growth, and then on the x-axis is the time in hours. And this is in our control and our different doses of IAA. So if you first focus on the very light gray line, the control follicles after about 48 hours substantially grow in size, as do those from the 2 lower dose groups, the 2 micromolar and the 5 micromolar. However, when we get to the 10 micromolar or the 15 micromolar, there's a significant inhibition of follicle growth compared to control. Now, there are 2 reasons that follicles won't grow. One is that there could be something killing the follicles, causing them to undergo apoptosis, or there can be something inhibiting the cell cycle so that the cells in the follicle do not proliferate and do not grow. So we began to look at the mechanism by which IAA was decreasing the ability of follicles to grow in vitro. And one of the things that we looked at was the expression of factors that control whether follicles live or die. Factors that cause follicle to live are called anti-apoptotic factors, and factors that cause follicles to die are pro-apoptotic factors. And this is showing expression of these different factors in response to IAA after we've cultured them with either vehicle control or IAI. So if we look at our anti-apoptotic factors, IAA significantly decreased expression of both BCL2 and BCL2L10 compared to control. And if we look at pro-apoptotic factors, IAA significantly increased expression of BAX and AFM1 compared to control. Now, similarly, we looked at the ability of IAA to affect the cell cycle, because it's the cell cycle that regulates whether cells in the follicle proliferate, allowing the follicle to grow. And we looked at factors that are promoters of the cell cycle, thus they help with proliferation, and one factor that blocks cell proliferation. And overall, in general, when we looked at the promoters CCNE1, CCNB1, CCND2, we see that IAA significantly inhibits these cell cycle promoters compared to control. Now, one of the cell cycle promoters, CDK4, was slightly increased in IAA treatment compared to control. However, those slight increases were offset by very large increases in the expression of the cell cycle inhibitor CDKN1A in IAA treatment versus control. Now, in addition to looking at cell death and cell proliferation, we wanted to see whether IAA was affecting the ability of the follicles to make sex steroid hormones. And so what I'm showing you here in the graphs are different sex steroid hormones that are important for controlling reproduction, not just in mice, but also in humans. And what we see is that IAA exposure is increasing the levels of pregnenolone and progesterone compared to control, but decreasing the levels of androstenedione, testosterone, and estradiol compared to control. And I don't have time to show you all of the data, but if you look at the schematic that I've shown here, I'm showing you how these ovarian follicles make sex steroid hormones. So we have thecal cells that uptake cholesterol, convert cholesterol to pregnenolone, then progesterone and DHEA, then androstenedione and testosterone. And what we see and what I'm showing you here is that IAA is decreasing androstenedione and testosterone and increasing progesterone and pregnenolone. And from studies I'm not going to show you, the reason that these changes are happening is because IAA alters the expression and protein levels of things such as increasing STAR and CYP11A1, which lead to these increases in pregnenolone and progesterone. And IAA decreases expression of HSD3B and CYP17 alpha, which leads to lower androstenedione and testosterone. And IAA also affects the granulosa cells. It causes them to make less testosterone and less estradiol because it's inhibiting these enzymes CYP19A1 and HSD-B1. So what I've told you from in vitro studies is that the disinfection byproduct IAA increases apoptosis by increasing pro-apoptotic factors and decreasing anti-apoptotic factors, decreases the cell cycle or proliferation by affecting genes that control cell cycle, alters steroidogenesis leading to low levels of estrogens and androgens, and collectively, I think that these things are leading to inhibition of follicle growth. Now, this is a very nice story in vitro, but in vitro is not the same as a whole animal. And so we began to wonder what would happen in a whole mouse that was exposed to iodoacetic acid. And the hypothesis that we tested was that iodoacetic acid exposure affects female reproductive outcomes in vivo. Now, to test this hypothesis, we used adult CD1 mice that we dosed either with vehicle control or different environmentally relevant levels of iodoacetic acid in the drinking water. Then we monitored the mice for estrocyclicity. We collected their ovaries to look at gene expression, and we collected serum to measure hormone levels. So first I'll show you the data on estrocyclicity. Mice have 4-day estrus cycles, which consist of proestrus, estrus, metestrus, and diestrus. And in the mouse, metestrus and diestrus are physiologically very similar, so we often lump them together as one cycle stage. And this is showing the percentage of days that the mice spend in each one of these cycles, but depending on whether they were control or different doses of IAA. And what we found was that IAA significantly reduced the time the mice spent in proestrus compared to control, but it did not affect the time the mice spent in estrus, metestrus, or diestrus. Now, we looked in the in vivo studies at the same factors that control follicle life or death, anti-apoptotic or pro-apoptotic factors, as we did in vitro. And I'm showing here on the left the expression of anti-apoptotic factors in mice that were exposed to different doses of IAA in the drinking water, and we're looking in the whole ovary. And what we saw was that IAA did not affect expression of BCL2. However, the highest dose increased expression of BCL2-L10. When we look at pro-apoptotic or pro-death factors, we see that IAA exposure increased ovarian expression of BAX, decreased ovarian expression of BAC, and initially at a low dose, decreased expression of AMF1, but increased expression of AFM1 at a high dose. We also looked at cell cycle regulators, again, the same ones that we looked at in vitro. And I'm showing you here the cell cycle promoters and the cell cycle inhibitor. And what we saw was that the highest dose of IAA in the whole ovaries increased expression of CCNA2, CCNB1, and CCNE1 and CDK4 compared to control. It slightly decreased expression of CCND2 and did not change the inhibitor CDKN1A. When we examined circulating levels of hormones in these animals, we did not see any effects on progesterone or testosterone. However, like we observed in vitro, IAA significantly decreased androstenedione levels and estradiol levels. And if you look on the right here at the pathway, when we looked at gene expression of factors that make these different hormones, we found that IAA significantly inhibited expression of CYP19A1, which is a critical enzyme for making estradiol. So if we compare what we found in vitro versus in vivo, I'm showing a table here that lists the different factors we looked at, the specific genes, and if we have a down arrow, that means they both had a decrease with IAA. An up arrow means they both increased with IAA, and an arrow that goes sideways means they had no change in IAA. So when we look at this, we see that there were a lot of differences between the in vitro work versus the in vivo work. However, there were some consistent findings that both in vitro and in vivo, IAA exposure increased expression of the pro-apoptotic factor BAX, decreased expression of the cell cycle promoter CCND2, increased expression of CDK4, and both in vivo and in vitro, IAA decreased expression of CYP19A1, which is a critical enzyme for converting testosterone to estradiol. And then when we looked at the sex steroid hormone production in vitro versus circulating levels in vivo, we see that IAA treatment, both in vivo and in vitro, decreased androstenedione levels and estradiol levels. So we began to think about what is different between in vivo and in vitro, besides the fact that in vivo systems, you're going to have the hypothalamus, the pituitary, the liver, the kidneys, the GI tract, which can all influence the outcomes that we're looking at. Another big difference is when we were doing our gene expression studies in vitro, those were focused on gene expression in antral follicles. when we're doing the gene expression studies in vivo, those studies were focused on whole ovaries, which contain not just antral follicles, but interstitial tissue, corpora lutea, and smaller, more immature follicles. So we wanted to see what would happen if we dosed animals in vivo, but then just looked at gene expression in the ovarian antral follicle. And so we tested the hypothesis that iodoacetic acid exposure affects the transcriptome in ovarian antral follicles. So to do these experiments to test this hypothesis, we dosed female CD1 mice with IAA in the drinking water or vehicle control for 35 days. After dosing, we collected the ovaries, isolated the antral follicles, and then subjected the antral follicles to RNA sequencing analysis, followed by enrichment annotation analysis to look at the transcriptome in these isolated antral follicles compared to control. So I don't have time to show you all of the data, but I will tell you that IAA exposure altered expression of genes in these antral follicles in factors related to RNA processing, regulating angiogenesis. Like we saw in vitro, we saw a lot of the same changes to the cell cycle, mitotic cellular division, and cell division. We also saw that IAA exposure is affecting the PI3 kinase AKT signaling pathway, estrogen signaling, GnRH signaling, insulin signaling, and oxytocin signaling in these antral follicles. So what I've told you in vivo, if you've got an animal exposed to IAA, that IAA can either increase or decrease expression of factors that regulate apoptosis. It can either increase or decrease factors that regulate the cell cycle. It inhibits steroidogenesis, particularly circulating levels of androstenedione and estradiol. And it can alter the time animals spend in proestrus, and it can change transcription of genes in isolated antral follicles. So in conclusion, I hope that I've told you that something we don't think about often, this disinfection byproducts that are in water that we use every day, can have impacts on the female reproductive system. And I showed you that in vitro, it can affect apoptosis and the cell cycle, disrupt steroidogenesis, and in vivo, you can have effects on steroidogenesis. We have alterations in the ovarian transcriptome. In vitro, we see impairment of follicle growth, and in vivo, we see disruption of the estrous cycle. So I do think that IAA has a potential to be a reproductive toxicant, and that further studies need to look at this, not just in mice, but hopefully in humans, and be able to look at whether exposure to disinfection byproducts like IAA has some of these similar associations with human reproductive outcomes. And then lastly, I would like to acknowledge my wonderful collaborator, Dr. Michael Pleva, and members of my laboratory. In particular, I would like to acknowledge Dr. Andrea Gornsiorski, who was a Ph.D. student in the laboratory, completed most of this work, and is now a resident at Michigan State University. And I would like to thank the funding and all of you. And I'm happy to answer questions later.
Well, thank you. Thank you, Dr. Flaws, for that amazing overview of this fascinating work and research. That's incredible. Reminder to everyone that we will take questions if there's time after our next speaker on this topic and session. Please place all your questions in the chat, and we'll review them after we hear from Dr. Natasha Mishkanovska. Sorry about messing your name up there, Dr. Mishkanovska.
Okay. It's all good.
I appreciate that. Just a little introduction on you before we get started.
Super brief, yeah.
Yeah, you're an incredible speaker and researcher, and that you were at the Mayo Clinic and Cleveland Clinic trained dermatologist and dermopathologist who currently serves as the vice chair for clinical and clinic trained dermatologists at the Department of Dermatology, University of California, Irvine. You have a busy research unit and dedicated to alopecia and aging and inflammatory disorders with your research. You're the director for integrative dermatology and at the University of California, Irvine, and the Integrative Health Institute for advancing evidence-based research in that area. As a faculty member of the University of California, Beckman Laser Institute, you're involved in developing non-invasive imaging modalities for skin and hair conditions. As an educator and a mentor, you've received many grants and awards for your clinical, educational, and research. You serve as Chief Scientific Officer for the National Alopecia Areata Foundation between 2015 and 2022, and essential in developing clinical trials that have led to novel therapies. You currently serve on the NAAF Scientific Advisory Board and the Board of Directors for the American Hair Research Society. Thank you for your time today and your great graciousness.
Thank you so much for the intro. We have 10 minutes, guys, so let's go through this, and I'm going to try not to get into details. I just wanted to show you, because when I was on the East Coast, I did not know where University of California, Irvine is. It's one of the newer things. It's down in Orange County, as in the Housewives of Orange County. It's beautiful. You have to come visit. Why are dermatologists talking to you today? Listen, we all know skin is the largest organ, and that is not a joke. It's out there and it's subject to the environment, right? One thing that I think nobody realizes is that bad skin equals bad health. And this whole new thing, something that I worry about and study, and the new wave of biohackers, the whole longevity science, there's this whole term of inflammaging, meaning inflammation is something that kind of leads you to age. Well, guess what happens to skin as we grow up? Skin composition changes, and it's around 45 years old, right? That's kind of like the grown-up limit for all of us. As the composition of the skin changes, the skin becomes more permeable, more porous, becomes drier, more itchy, and more prone to inflammation. So guess what happens with inflammation for the skin? It does enter your body's circulation. So just base sheer on its size, inflamed skin contributes to whole body inflammation and aging. So when people ask, do you have to use moisturizers? I'm always like, there's beautiful remote parts of the world where people never have to put anything on their skin. But, actually the joke is on me, because the truth is studies that have looked at grown-up humans, that I did not put this word, that's what the authors used in the study. When you use skin emollients daily, so you keep the integrity of the skin when you're more adult, it actually lowers the inflammation markers in your blood. So see how little kind of emollient on the skin translates into systemic health. So skin health is overall health. The second thing that we dermatologists have to deal with is really the skin cancer is the #1 cancer. You're going to look at statistics and you're going to say breast cancer is #1 in women, and then down on the bottom of the table is going to be like non-melanoma skin cancers excluded. What does this mean? This means that we have to not only diagnose and prevent, but we have to teach people how to protect themselves. And because people don't want to stay inside all the time, because we need vitamin D, as we heard earlier, we have to instruct people on what to use in their skin and how to use that even at a young age. So we have to be very cognizant of what are we putting on the skin and how is that going to affect people of all ages. And the third and the most relevant thing is really that nowadays, anyone is a skin expert. You're all on social media, whether it's a dancing doctor on TikTok that just graduated from residency and is not really seeing patients, or a 13-year-old telling your kids what to buy. We all subject ourselves and buy all sorts of stuff, not knowing where to go, because skin, hair, anything that we do is a big market with a lot of snake oil. So how do I choose what to talk about today? Because there's so many things. I'm going to tell you about 2 things, and quick. These are things that come from my clinics and from my patients. So one is the new alopecia epidemic, and the second one, if we have time, the banned sunscreens. For me, 2020, when the world stood still, when the whole COVID thing happened, was probably the biggest exocrine and endocrine disruptor in my life. But one thing that I learned, I think we all learned from the whole COVID, was really for all of us that got COVID, and some of us even with the vaccines, was the prevalence of hair loss. So we learned a lot about hair not wanting to simply through COVID and the inflammation and the long terms things. But one of the things I'm going to talk about today is really something that started a little bit before COVID, and it's called the new epidemic of this condition called frontal fibrosing alopecia. For short, we're going to call it FFA for the rest of the talk. This is a scarring type of alopecia. What does that mean? When the hair falls out, which alopecia just means hair loss, the skin area starts looking like a scar. There's no little root, so it cannot come back. It's a whole family of a lot of rare conditions. This is from a chapter, so tons of them exist, but these are the most common ones. And actually, this FFA, frontal fibrosing alopecia, is a subset of this thing called lichen planopilaris. I know, long words, but what they pretty much tell you is people start getting this red scalp that's itchy, a little bit scaly, and then the hair starts falling off and it cannot grow back. Frontal fibrosing alopecia is a very particular thing because it got its name from these foreheads kind of going up and pushing the hairline back to the sides and even on the back. The follicles are completely destroyed and we don't have a way to regrow them back. One thing we know is that it affects postmenopausal women predominantly, this being the average age, but the youngest range keeps expanding every year, and the last couple of years there's even kids cases. The interesting thing is that this as a disease did not really exist before the 1990s. So all of my professors, when we go to these national, international meetings, they go through their stacks of photos. They really didn't see any of this. So everybody trying to figure out what is it that happened in the 90s? Was there something that entered the market then? So these women will be like that? And some of the things are like a lot of skin preservatives and sunscreens that are chemical that entered. So this is all going to tie into a story. And the interesting stuff is that every year we see more and more cases and the lower age expanding. What women tend to have is a lot of them tend to have rosacea, or this redness of the face. The thing that tends to precede the hair loss will be eyebrow loss. So you can see in this patient how the eyebrows are gone. It can be very minimal, like just right here, kind of the forehead is expanding, but it can also just keep pushing. On the one side, you see a little bit of redness, and then when the redness goes away, the follicles are killed and that stuff will not come back. One good thing, or one way we can recognize these patients, is because they don't have many wrinkles. So the only good thing, here's a person with stuff on the side, which kind of turns white, or they can have it centrally. Again, loss of that pigment. The veins become prominent. So here is a lady in her 50s and a lady in her 70s, just with these prominent veins. And it's called frontal, but it also starts wrapping around the back. So a lot of women will have loss of the hair in the back as well. In people of darker skin color, you can actually have this forehead pigmentation that can be prominent, and on the face, almost like a thing called melasma. And in many patients now, we're seeing almost this cobblestone bumpy structure of the skin, and we refer to them as facial papules, something that's very challenging. So back in the days, we used to think, this happens for a couple of years and then it stops. But now we know it doesn't really. It goes through waxes and wanes, and it can actually be associated with loss of hair on the entire body. That one is usually not scarring, but it's present. So what do we know about this condition? We know that it's autoimmune. Some of the pathways have been decided and discussed, but there are no blood markers or way to test for it. It's a huge treatment challenge because first you have to control that inflammation, and then you have to try to regrow the hair. There are many, many algorithms of how to treat these and which way to go. But bottom line is this. Here is an example of a patient who's red and inflamed. You can calm him down with medication, with like maybe injecting platelet-rich plasma, but then it will just come back because it's a chronic condition. This is the extent of stuff you can maybe regrow with all attempts in very young patients early, but in people that are a little bit more adult, it's just very difficult to get anything. We get excited over 2 hairs. Here's another patient. We got a little bit of hair, just a rest, but it can kind of take off at any time. The facial papules, we now use vitamin A or Accutane to kind of calm them down and smooth them out. But as you can tell, it still doesn't affect the hairline or regrows the hair back. So the thing we're trying to figure out is how to stop this thing, because we can't treat it really. So how do we stop it? There's this theory that this is all related to some cell-mediated allergic response. What my group figured out from studies that were big and other groups have confirmed it is that there's a very high association with thyroid disease. There's also associations with low androgen level and low steroids. But one of the things that keeps popping up is really allergy, contact dermatitis, and the role of sunscreens. So what is the role of sunscreens? A very large study, a questionnaire, 2016, that was done in the British health system, figured out association between this condition and people that were using, so high use in these areas of the world, facial skincare products that were leave-on, so not something you wash on. So like moisturizers with sunscreens, and sunscreens. For example, men, 71% of them that had FFA were using sunscreens twice a week year-round versus the ones that did not have the disease. Parts of the world like Brazil are plagued by this condition, so they tried to see if in their population they have this stuff, and they found that, yeah, moisturizers really also were one of the associations that they saw there. They also saw maybe the use of hair straightening with formalin, but one thing that they showed is maybe the clarifying shampoos will help, but the sunscreen thing was really not strongly found. The latest meta-analysis from all the things, it still rings the same. There is this association, you cannot say causality with facial sunscreens, doesn't say which ones wear what, and facial moisturizers, particularly for males. And all of these have something that is UV protecting chemicals. And UV protecting chemicals nowadays can be in anything from shampoos to even heat protections that I use to straighten my hair. No significant relationship has been found in any of the cleansers, toners, foundations, or hair products such as the shampoos, conditioners, mousses, and hair gel. Contact allergies are also kind of tied into this as we're trying to figure out the role of the sunscreens. We know that a lot of the women will be allergic to nickel and things that are found in shampoos, primarily of the fragrance and natural things. So that's why natural sometimes when it comes to skin products can be a really, really strong source of irritation. And then the whole formaldehyde that we discussed. The role of sunscreen. So this is a picture of a patient of mine who put a sunscreen, and you can see he has FFA on his scalp, but he showed me a picture of his arm where you can see the little bumps showing up on his arms. And he was a very popular brand of a chemical sold, sunscreen sold in Sephora. What are the hypotheses of how these sunscreens are doing this? There are many, many hypotheses. One is that sunscreen enters the follicle and there is an immune reaction. For example, stuff has been shown, the deposition of titanium oxide, which is not chemical, it's a metal. But also there may be, it's not just the metal on its own or the chemical on its own. It can also sensitize the skin to other things that irritate it, such as all these things called preservatives like methyl isothiolisonone. The other thing is why postmenopausal women, is it affecting something endocrine? Is there sebum production? So a lot of unknowns still left there. One of the interesting things that came out of Australia, where a lot of people use sunscreens and have great hair experts, is a case in a younger patient where she had this condition, FFA, and it reversed after she stopped using sunscreens on the forehead area. So this led the authors to say the following. We now recommend that patients with this condition avoid applying sunscreen products on the forehead and use a cap or hat for sun protection. Now that sounds easy, but do you remember what the #1 cancer in the world is? And that's my job, right? So how do we navigate this and how do we go through this? Which sunscreen is it? We don't know. Chemical are the ones because of the 1990s that we're thinking. Physical, the titanium may be a reason. Zinc, nothing bad yet. So that's probably something I stick with after doing all these things. Evidence on the sunscreens and everything still needs to be interpreted with caution, but I will tell you some of the things that we kind of looked into. So the banned sunscreen ingredients and the impact on human health. The banned two sunscreens in 2018 in Hawaii are these things, oxybenzone and octinoxate. These are the names, BP3 and OMC. Why? They were purportedly bleaching corals. So why did we look into them? Because patients started coming, and all my questions always come from patients. And they're like, what is this doing to my body if it's really bleaching corals? And I'm like, I honestly don't know what it's doing to your body. But a study did come out several years ago saying, listen, stuff you put in the skin is in your blood in a couple of hours. So actually, the absorption of these sunscreens is shown after whole body application within 2 hours, and it can linger for several days. So patients and we are, justified to ask what's going on and why is it going on. So we looked at all the stuff that's out there for these sunscreens and being endocrine disruptors, because they're very common sunscreen ingredients. And animal and in vitro stuff, like we just heard, really had a lot of things to show that it does something in experimental models, but what does it do in human models? So the article that we published really tried to assess what's going on out there when it comes to human health. These two things are not just in sunscreens, though. You find them in air, the drinking water we just discussed, and they also protect fragrances, cosmetics, plastic packaging. So they have many ways to get into the skin, which means we have a lot of ways to also learn about their effects on us and what are their negative consequences. What is the story with oxybenzone? It's actually a naturally occurring in flowering plants. It absorbs and scatters the sun's UV rays. That's why it's used. In sunscreens, it's usually less than 10% presence. It's also in many cosmetics. It doesn't let things go bad and rot. So it's a lot of plastics, a lot of toys and furniture. Testing usually is done in the urine, and the exposure is really notable in the United States. Actin oxide, not much, because it has a less skin penetration, so not much is known about it in systemic absorption. It is present in cosmetic products. And the thing is that from cosmetic products, it's still the amount that it enters in the body is above the FDA toxicology threshold. So we need to know about it and we need to learn about it. The results is that we found 29 studies that met criteria on these things, these two products being internal, only 2 for OMC. 16 were for organ health, and 13 actually were prenatal exposures and what happens to the kids. So let's go over these things super quick. BP3 was found in a lot of urine samples, especially in North America. And apparently, urinary BP3 in North Americans are 10 to 20 times higher than Europeans and Asians. So we're very exposed to it. Is our microwaves? Is our plastic? I don't know. Sunscreens are used widely across the world, so I don't think sunscreens are the cause. Thyroid association is the one that's been studied in most detail, but still it's inconclusive because some studies say there's association and some studies say no. The initial one said that there was association with decreased thyroid hormones with high BP3. However, the most recent study, which was big, over 1,000 people, did not find any statistical significance. So yes, we're going to be cautious, but hopefully we'll get a little bit more answer. Also, no connection of OMC with thyroid level. When it came to reproductive hormones, there are studies on both the estrogen and androgen activities. In vitro and animal studies, a lot of activity in this area. However, in women, there was nothing that was found in studies. In men, the story is a little bit different. The association that was found with lower testosterone was in adolescents. So it's our puberty kids. And then a little bit in older, but it was still found to be within the normal physiologic age. There was no effect, though, still in these men on fertility or the quality of their semen. When it came to disrupting potential for puberty onset, there are still studies that, again, were up and down. So they're contradictory in association, but still we're paying attention because now there's a couple of studies for this potential being present for pubertal onset, particularly in boys. Impact on fertility, there was none. So we can relax from that, and I'm going to skip that. And for kidney function, there was definitely an association between kidney function and cumulative renal injury. And this is from all over levels of BP3, not from sunscreen necessarily, but just to tell you that some of these parameters in kidney functions can be affected with long-term exposure, and we're yet to find out whether applying it on the skin versus absorbing it from everywhere else will affect us. For obesity, childhood obesity, urinary concentration is something that's been looked at, but there's no significant association. So this is the summary of these 2 banned sunscreens and what we found out. There's a signal probably for thyroid hormone, just because there's different studies, the male reproductive hormone level, particularly adolescents, and the kidney function. There was no issues with OMC, so we can rest relaxed about that one. When it came to fetal and neonatal development, because the embryo and the fetus are vulnerable, there's sunscreen concerns for use in pregnancy. So usually all the studies really will follow up the babies after birth and check the mom's urine during pregnancy. For OMC, there's nothing that's been studied, so I can't even comment. This is what they found out: possible offspring defects and birth weight with the BP3, but not with OMC. So bottom line, in our study, we did not find a strong support for causal relationship within systemic levels, but there are questions in thyroid, reproductive hormone levels, puberty development, and kidney function for BP3. For OCM, that's what it is. So I just want to thank you for your attention. These are our discussion points, and I think that was the fastest I've ever talked in my life. Okay, have a great day.
Thank you so much, Dr. Mishin-Kowalska. I'm actually very impressed. You've got an awful lot of information in a very, very narrow window. We'll take time for a couple of questions. Dr. Flaws, there's a couple questions for you. Do you know if disinfection byproduct exposure is concentrated in any particular region or population subgroup, or is it fairly uniform?
So I would say that all groups and populations are exposed to disinfection water byproducts, but the type of byproduct and the type of disinfectants used in different areas differs dramatically. So in Illinois, for example, I may not be exposed to the same ones as somebody living in China or India or other parts of the country. So I think there's ubiquitous exposure, but the types of exposure are going to differ depending on how the particular municipality disinfects the water.
Thank you for that. Have you identified different, is there a difference in chemical levels between tap and bottled water?
So I haven't done those kind of studies myself, but others have, and there are differences in the types of disinfection byproducts in tap versus bottled water, but both tap and bottled water have them. So it's not like bottled water eliminates them.
Okay. And Dr. Mission-Kavoska, did you see any consideration for the level of melanin in groups or populations globally?
I assume this is for FFA. We thought we see it a little bit less in darker skin color. Some of the photos that I showed you, actually the patients that I showed you are patients that are using sunscreen. So one of the patients is a male person. She distributes mail, so she uses sunscreen every day. That's why the connection. But from South America, from Brazil, there are definitely patients with skin type 4 and 5. They're predominant kind of epidemiologic group. So we know, although we thought it was rare or much more rare, we're definitely seeing it, and particularly in our Hispanic population.
Very good. Thank you for that. And there are several more questions in the chat, and if you would both, would not mind responding to them, we'll, we're short on time, so we'll just, and thank you for working so hard to get us caught up. Thank you. We'll shift gears to our next session, outlining endocrine disrupting chemicals and breast cancer risk, and honored to introduce our next set of speakers, Dr. Elizabeth Boham and Dr. Michelle Lamero, and Dr. Lindsay Trevino. So we'll start off with Dr. Boham. Dr. Boham is a physician and nutritionist who practices functional medicine, is the medical director of the Ultra Wellness Center in Lenox. Dr. Boham completed her undergraduate degree in nutritional biochemistry at Cornell University and her master's degree in nutrition and registered dietitian at Columbia University Teachers College. She then went on to medical school and is a board certified physician in family medicine. Dr. Boham has developed a functional nutrition course that is being used to educate physicians and other health professionals worldwide, and has contributed to many podcasts and articles, and wrote the chapter on obesity for the 9th edition of the Rankel textbook on family medicine. Dr. Boham was diagnosed with triple-negative breast cancer at the age of 30 and has worked to teach other women what they can do to decrease their risk of breast cancer through lecturing and her breast wellness DVD. Through her practice and lecturing, she has helped thousands of people achieve their goals in optimal health and wellness. Dr. Boham, the floor is yours.
Thank you, Deb. Thank you so much. And really, it is an honor to be with you all today. It's amazing that we're having this symposium. I'm really honored to be asked to be part of it. And as a clinician, it's just wonderful to be with all these amazing researchers doing the really important work to get this knowledge, this research done and get the information out there. And as Deb mentioned, I am interested, I'm board certified in family medicine. I do functional medicine, which is just, a way of thinking about health and the body and how all the different systems in the body are interrelated. And when I was diagnosed 24 years ago, I thought I was the healthy one, right? I was all interested in nutrition and exercise and prevention. I didn't have a family history of breast cancer or much history of cancer of any sort in my family at that point. And so, when that happened to me at such a young age, and as a clinician, as a physician, I was asking that question, why? What's going on? And one of the things that really I thought a lot about was how we were collecting data on women and doing this research. And I was thinking about, we're missing the boat here, because so often when we're looking at cancer clusters, we're thinking about where women are at the time of diagnosis. And as we're learning today, as we've gotten such great information earlier today, really, of course, what's happening in when we're a fetus early in life, during puberty, is really impacting our risk of this disease way longer, as we get older in life. And so that's important. I think that endocrine disrupting chemicals are really bringing that to light, and I'm really happy to be part of this. So we're going to go over I'm not going to spend too much time on endocrine disrupting chemicals, but also talk a little bit about not only how can we avoid them, but phytonutrients, how they can impact, how much these chemicals impact our health, and what you can do to decrease your risk, and what we can do on a more global way. So we know, the statistics are grim. They're significant. About 1 in 8 women in their lifetime will have an invasive breast cancer. 1 in 39 or 3% will die of the disease. These are the numbers from 2022, where we had over 287,000 women being diagnosed with an invasive ductal breast cancer or an invasive breast cancer, with an additional DCIS on top of it. And 4.1 million women are living with breast cancer in the US as of today. So, and unfortunately, this incidence continues to increase. And what's also we're seeing is, even though we see here the median age is 62, we're seeing younger and younger women getting diagnosed. When I was diagnosed at 30, 24 years ago, it was rare. I was the rare one. I didn't know anybody that was less than 30 at the time diagnosed. And now, unfortunately, I take care of women in their 20s who are going through treatment. And so we know from that age group of 15 to 39, of this 287,000 women, about 70,000 women are in that young age group, that 15 to 39, and that has been increasing. And for those women, it's the most common cause of cancer. For all women in general, it's the second most common cause of cancer after skin cancer. We know that the cause of breast cancer is multifactorial, right? There's a lot of things going on that's influencing a woman's risk, from her genetics, both high and low penetrance genes. So the high penetrance genes are things like the BRCA variation, and low penetrance genes would be genes that multiple women have variations in that do impact our risk, from COMT and methylation genes to glutathione genes. And we know our reproductive history impacts our risk, our lifestyle, of course, obesity, diet, physical activity, alcohol intake. But we also know that environmental exposure really increases our risk. And that's what we're really focusing on here today. And since World War II, there's just been an increase, as we saw with Dr. Gore earlier, that there's been an increase in disease risk with an increase in chemical exposure. And we are seeing that statistic with breast cancer as well. We know there's over 95,000 chemicals, both industrial and agricultural chemicals, that are approved or are on the market in the US for use. And very few of those have been adequately studied in terms of toxicity and health on people. And what was it, since 1976, that Toxic Substance Control Act was passed, the United States has only eliminated or taken off the market 5 chemicals. So I wanted to divide this talk into endocrine disrupting chemicals, what matters, right? So what matters in terms of how do they impact our risk and our health? So we know exposure, of course, matters. The timing of that exposure matters, our genetics, and our lifestyle. So let's talk about exposure first. I thought this was an interesting article or drawing that showed about all these factors that can impact a woman's risk of breast cancer. And it really detailed all the different endocrine disrupting chemicals, as well as other chemicals and hormones and lifestyle a little bit on there too. So as mentioned earlier, these endocrine disrupting chemicals are impacting hormone action in multiple different ways. And a couple things I wanted to point out, though I know you've heard a lot about this today, but BPA, think about those thermal receipts that you receive when you're, checking out at a store. And of course, the lining of food cans, as well as the hard plastic water bottles, and it sneaks in there. And so phthalates, think about the fragrances added to products, both cleaning products as well as personal care products. Parabens, I'm always checking my moisturizers, my makeups for any of these parabens, methylparaben, propylparaben. But, you know, it's also unfortunately in our food supply, especially the ultra processed foods. But I was looking at a package, not that I wanted to buy these, but they were not, you know, they were a package of soft tortillas. And when you look at the ingredients, it was an ultra processed food. There was multiple ingredients, but there was a paraben in there. So you can find parabens in your food supply in some of these different ultra processed muffins, tortillas, for example. Triclosan was banned from antimicrobial soaps in 2016, but still in our as an antimicrobial in toothpaste, personal care products as well. So BPA, you know, as you've learned a lot about today, with all that amazing research that's been shared, we know that exposure in rats can increase the number of precancerous lesions that will then decrease the threshold that a carcinogen can impact that precancerous lesion later on in life, in the lifespan. Neonatal exposure of mice to BPA increases the sensitivity of estradiol when they're going through puberty and in their mammary gland production or growth. BPA can impact people epigenetically, as we've been talking about. It also can reduce the efficacy of certain chemotherapeutic agents. But interestingly, a 3-day period of limiting intake of packaged food decreases the concentration of BPA found in the urine of people by 65%. Phthalates, you know, can bind to the estrogen receptor. They can also impact proliferation of malignant invasion and tumor formation, even on estrogen receptor negative breast cancers. They also can interfere with the anti-tumor action of tamoxifen. In addition, though, if a person has a limited intake of processed food for 3 days, we see less phthalate levels in their urine. We've been talking a lot about other chemicals today, like the forever chemicals, the PCBs, atrazine, DDT. So timing, I think this is so interesting, as I was mentioning earlier, and I think what we know with some of these endocrine disrupting chemicals is that when we're exposed at a very critical time of development, prenatal, puberty, that can have an impact on our risk of disease later in life, right? And there can be a long latency between exposure and diagnosis, which of course is important for paying attention to when we're doing epidemiological research. And there's not this linear relationship with dose of toxin and outcome like there is with some other toxins because it's disrupting the endocrine system. So there's not that typical linear relationship always. BPA has had the most profound impact in early development. We know that BPA exposure can change the mammary tissue and the risk for that mammary tissue developing a tumor later in life. You've heard about DES today and how it can impact risk of breast cancer and how that can be for multiple reasons and possibly through the epigenetically. Here they talked about DDT exposure in the mother and how that can increase risk of breast cancer in the daughter at the age of 52, right? So a really long latency. So genetics, I spoke a little bit earlier about both those high penetrance genes that we focus so much on, the BRCA gene variation, for example, but then there's more and more research being done also on these low penetrance genes that lots of us have variations in. And when they come together in a certain combination or when the people have multiple low penetrance genes, that can impact a person's risk and how they handle toxins from the environment. But let's delve into lifestyle. Because this is what I like to talk about, even though, you know, sometimes, like we've mentioned earlier, it's hard to always find out, you know, always know what we can do here. But this is kind of interesting. This looks at these toxic plastic compounds like BPA and how they can cause many different disorders in the body. They can be neurotoxic, immunotoxic, you know, damage the immune system, damage, you know, gonadotoxic, damage our hormone system, carcinogenic. But this study is looking at how polyphenols from our plant foods can actually block these negative impacts of these endocrine disrupting chemicals. And so let's back up for a second. Polyphenols, right? Polyphenols are these plant compounds like they're phytonutrients, right? So in foods, we have the macronutrients, which is your protein, carbohydrates, and fats. You have the micronutrients, which are your vitamins and minerals. But also in our plant foods, we have these phytonutrients, right? These are these other nutrients in our plant foods that have this tremendous ability to impact our health on so many different ways, right? We've seen certain phytonutrients have an impact to lower inflammation in the body, and certain phytonutrients have been shown to decrease the carcinogenic potential of something here. And so this study is really looking at how these polyphenols, these phytonutrients, can block these negative impacts of these plastic compounds and make it so that they're not as immunosuppressive or not as carcinogenic. And we know that these toxic compounds from plastics are dangerous to us as well as the environment. They can impact oxidative stress in our body and inflammation and promote cancer progression and metastases. But this study has shown that these polyphenols can inhibit cancer progression and metastases. And here just showing on the cellular level, both in a cancer cell as well as a normal cell, how these green peas, polyphenols, can block the negative impact of, for example, here, BPA. So as I was mentioning, these phytonutrients are these important components in our plant foods. They are there in plants to help the plant survive in nature. They help the plant defend against microbes in the environment. And so there's some thought that if a plant has to work harder to survive, that they may then contain more of these phytonutrients, and that maybe our organic produce or organic plant foods have higher doses of these phytonutrients. In addition, when we're choosing organic, we are avoiding more of that herbicide, pesticide exposure as well. But phytonutrients have been shown to reduce the oncogenic potential of carcinogens. And as you see here, we are working to eat the rainbow and get our plant foods from all sorts of different colors of the rainbow, so we can get a wide variety of these phytonutrients that can have really beneficial effect on our overall health and cancer risk, right? So for example, some phytonutrients include chlorophyll that you see in your green vegetables, or the glucosinolates in your broccoli or broccoli sprouts or cruciferous vegetables, quercetin in your onions, or ellagic acid in pomegranate. And the goal, what I always tell people, your goal is 8 to 10 1/2 cup servings of vegetables and fruits a day. And that's the typical serving. When you say 1/2 cup, is if it's not cooked. But when we're talking about phytonutrients, we're saying 8 to 12 servings in a day. And the reason why there's a higher goal for phytonutrients in general is because the other things that fit into phytonutrients include your spices, which are a wonderful source of phytonutrients for people, and your teas and your coffees. So in addition to all your vegetables and your fruits, your spices and teas and coffees are where you're going to get your phytonutrients. And your goal is to really get a wide diversity. You want to get a wide diversity of different colorful plant foods in every day. Try to get something that's red and orange and yellow and green and purple and tan, right? And get a wide diversity, mix it up, and get a large amount, right? Isoflavones, these are also phytonutrients. Genistein, diazine, equal is made in our gut when we have healthy, good gut bacteria. These phytonutrients are considered phytoestrogen, right? They can actually impact the estrogen receptor. And what has been felt when we are looking at these foods from a food source, right, is that they have weak estrogenic activity, and they may be blocking or antagonizing the impact of our own body's estrogen, or maybe other toxins from the environment that impact our own body's estrogen. So it's been felt that things like lignans, which include ground flaxseed, for example, or soy, will actually lowers a woman's risk of breast cancer because of that. Ground flaxseed is wonderful because it's also rich in omega-3 fats, which lowers inflammation in the body. People ask me about soy all the time, and this is a good study that came out looking at women at high risk for breast cancer and soy intake. And they found that when women eat, whether they had a history of an estrogen receptor positive or estrogen receptor negative breast cancer, they were on tamoxifen or not on tamoxifen, that soy was a decreased risk of death and recurrence and was safe and may actually be beneficial. But I think what we need to really step back on and talk about here is this is when we're talking about soy as a healthy food source and ground flaxseed, right? So I'm encouraging, I think it's fine to consume organic soy in a whole food source like edamame and tofu and tempeh. I love ground flaxseed. I think there is big questions, like Dr. Williams brought up earlier, with regards to soy as a formula for our infants. I think there's a lot of questions that need to be looked at there. And in addition, I always caution women to avoid the concentrated soy products, like things that have soy protein isolates in it. You know, they add this to bars and cereals to increase the amount of protein in that food. That I don't feel has been adequately studied. And I do have some concerns with that concentration in those forms. And I also have women avoid soy protein powders. I think there's a lot of other options there, and that's just not adequately studied as well. So what can you do to lower your toxic load, right? We want to choose organic. We know that kids, for example, that are eating plant foods that are organic or getting most of their foods from an organic source will have 1/6 of the amount of herbicides and pesticides in their urine as kids not choosing or not getting most of their food from organic sources. In addition, when you're choosing organic with your animal protein, your red meats, you're going to decrease your growth hormone exposure. You want to be choosing glass whenever possible, glass to store your food. You don't want to heat in plastic, of course. You want to just avoid plastic, and, you know, something says it's BPA free. There's just some other chemical in there that's probably just, you know, not safe either. So really choosing glass. Limiting your processed and packaged food, as we saw, that had a huge impact on our endocrine disrupting levels, chemical levels in our urine. Cook at home more, right? You're going to just limit the amount of packaging that you're going to have exposure to. And you're going to be getting a diet in general that's more nutrient dense, more whole food, richer in fiber and phytonutrients that probably can block some of the negative impact of these toxins we're exposed to in our environment as well. Switch to glass or stainless steel reusable water bottles. Read labels on your personal care products. Choose unscented products, both personal care products and cleaning products. Avoid herbicides and pesticides on your lawn and garden. And, you know, this is really these are a couple of really great resources. Oops, sorry, went too fast there. A couple of websites, or Environmental Working Group and Think Dirty app, if you want, you know, to look up a product. But I want to think a little more globally too, and at a higher scale, because this is important that, you know, we want to work on this from a much larger space here. And what we can do is we need to work to collect data on whole life exposure to these endocrine disrupting chemicals. We need to move away from packaging containing these endocrine disrupting chemicals in favor of more biodegradable products. We want to apply some policies that can restrict endocrine disrupting chemical use in our environment. And I work with Mark Hyman at the Ultra Wellness Center in Lenox, Massachusetts, and he has a big initiative that's Food Fix campaign, where he's working to transform federal dollars that are used to support food as medicine, working to incentivize regenerative agriculture, organic agriculture, reimagine our agricultural practices, our nutrition and our health, and improve the food supply so that more people have access to healthful foods. Because what we want to do is we want to both lower our endocrine disrupting chemical exposure as well as improve our food supply, so that's going to help with the health, our own health, as well as the health of the planet. So thank you so much for having me. Really, it's been a pleasure.
Thank you so much, Dr. Bowman. Thank you for that presentation. Continuing the conversation on EDCs and breast cancer risks will be Dr. Michelle Lamerle, who earned her Ph.D. in toxicology from the University of North Carolina, Chapel Hill, and her MPH in epidemiology while training as a postdoctoral fellow in physiology at Mount Sinai School of Medicine. Dr. Lamero is a professor in environmental toxicology and a member of the Environmental Health Center, Comprehensive Cancer Center, and Genome Center at the University of California, Davis. She is also a shared faculty member in the Environmental Genomics and Systems Biology Division at Lawrence Berkeley, and uses cellular, whole animal, and human epidemiological studies to identify physiological and molecular mechanisms of toxic exposures to humans. Her particular focus is on the relationships between developmental exposure and environmental pollution and the risks of endocrine diseases like obesity, diabetes, and breast cancer. Dr. Lamero served on the International Agency for Research on Cancer monograph that led to the international group of experts who published a consensus statement on the characteristics of endocrine disrupting chemicals in Nature Reviews Endocrinology. Dr. Merrill, the floor is yours.
Thank you. Just setting the timer to stay on track. Okay. So thank you for the invitation. There's been a lot of great presentations that I think really help set the stage for some of the material I'll be covering today, particularly Dr. Bohem. And so I'll go ahead and get started. So I do want to talk a little bit to you about IARC, which was just mentioned in my introduction. So IARC stands for the International Agency for Research on Cancer, and they're really the world's authority on carcinogens, so identifying these chemicals that have the property of being able to cause cancer. And IARC is part of the WHO. What they do is they classify the carcinogens based on available scientific information, using a group of international experts that don't have a conflict of interest. When they finish their work, it's written up in something called a monograph. And sorry, I got distracted by the chat, excuse me. The classification is based on the weight of evidence, where human evidence is weighted most heavily. So you'll see this in a moment. Their classification framework is pretty methodical. So what they do is they have these experts in evidence for cancer in humans, evidence for cancer in experimental animals, and then another group that reviews the evidence that's considered mechanistic or molecular. And so at the end of the meeting, the monograph will describe an agent as group 1, group 2A, group 2B, or group 3. And if something is considered to have sufficient evidence of cancer in humans, then basically the other two groups don't even need to speak at that monograph. It automatically becomes group 1. But I have this arrow on the next line because what we often find is more the case for group 1s is that there's limited or inadequate evidence in humans because of the difficulty of doing such work, and that we rely on evidence of cancer in experimental studies as well as strong evidence of mechanistic, and particularly in exposed humans. And so you can see with this matrix that there's different combinations that lead to these groups. And I think it's helpful for all of us to understand how that process works because of the importance of the IARC classifications. So sometimes I've heard people skeptically say, well, what, does everything cause cancer? And the reality is no, that's not the case. So when we look at the first 131 monographs of IARC, you can see that most of the agents that have been reviewed were called group 3, where there was not classifiable as to its carcinogenicity in humans, meaning basically there wasn't enough information to make a call either way. And so carcinogenic to humans included at this time about 121 agents. Group 2A can also be influential on the world stage. where it's probably carcinogenic, and we've got another 93 there. So just to give you a sense of what some of these group 1 human carcinogens are from IARC, you can see on the left that the agents that they consider are not just chemicals, but also occupational exposures and complex mixtures of chemicals, like the classic tobacco smoke. So I'm just highlighting that some of these chemicals include estrogens and diethylstilbestrol, which is usually referred to as DES. And when you look up specifically this convenient table that IARC put together, where they look at sufficient or limited evidence in humans per cancer site, and you look at the part of the table that is focused on breast cancer, these are the listings. So there's sufficient evidence in humans, according to IARC, for alcoholic beverages, DES, estrogen, progesterone, contraceptives, and menopausal therapy, as well as some radiations. I've only heard a little bit about some of these in the previous talks. And then with limited evidence, we've got some pesticides. Ethylene oxide is a sterilant that is used to sterilize medical devices and specialized equipment that can be used in high heat sterilization, night shift work, polychlorine, PCBs, as well as the tobacco smoking itself. So one of the things about breast cancer is that it seems more or less that all the known risk factors for breast cancer are linked to total lifetime exposure to estrogen. And I put a caveat because radiation is a very well-established environmental cause of breast cancer in women, but it doesn't have a clear link to estrogen. However, early puberty is known as a risk factor, which means your total lifetime of exposure to estrogen would be increased as similar on the other end of that, late menopause. The excess body fat relationship that we heard about earlier is believed, at least in part, due to the fact that fat cells actually produce or make estrogen. That link with alcohol that we heard about is surprisingly related to estrogen, and that alcohol can actually increase estrogen levels. And DES pretends to be estrogen, so it's an endocrine disruptor. So when we think about estrogen, 75% of human breast cancer is ER positive. And when we say that, what we mean is the estrogen receptor. So it's just an abbreviation. And these cancer cells are actually carrying the estrogen receptor. They could potentially not have the estrogen receptor, and there's a fair bit of, as you can tell, 25% of human breast cancers that don't have it. But when they have it, what happens is that the estrogen receptor acts like a lock, and the key that opens the lock could be an estrogen in your body, which I labeled natural estrogen in the blue diamond, or it could be something that mimics estrogen, like some type of endocrine disrupting chemical that acts like the key and also can open the lock. When the lock is opened, and what happens instead of a door being opened, for example, is that estrogen receptor and its key cause signaling in the cell. And ultimately that signaling leads to growth of the cancer cells and also the cells that help make blood vessels. And the blood vessels are really important for bringing nutrients into the cancer to help the cancer grow, and also potentially to help the cancer metastasize to distant sites. And of course, metastasis is very concerning because a cancer of the breast by itself wouldn't kill you because the breast isn't an essential organ for life. But if the cancer moves to a different part of your body that is essential, like your lungs, then this is a real concern for whether or not you'll survive. So, sorry, my slides got distorted. They were, let's see here. So diethyl stilbestrol, kind of a mouthful to say, so we usually say DES. It was used to promote fetal growth and particularly to prevent miscarriage back in the middle of the last century. And it is an estrogen mimicking endocrine disruptor, but as a pharmaceutical. So about 10 million women were thought to be exposed by, being prescribed this because miscarriage is, a great tragedy that it would be great to prevent if we could do so without harm. But in 1971, the FDA advised physicians to stop prescribing it because they realized that this rare vaginal cancer was arising in the female offspring that were born from women who took DES while pregnant. And so there's a number of reproductive risks associated with DES across the generations. We've heard previously about the importance of early life exposures and also potential for multi-generational exposures to cause harm. So one of the kind of classic case studies, if you will, a tragic natural experiment was with DES. And so the women who took the DES when they were pregnant, they themselves had an increased risk of breast cancer. Their daughters had not only increased risk of breast cancer, but also the reproductive tract, including those rare vaginal cancers that I just mentioned. They also had issues with structural defects in the reproductive tract, which is thought to be related to infertility issues and not being able to take a baby to to full term. And the sons of the women who were pregnant also had structural defects and infertility issues in their reproductive systems. The grandkids now are being followed. And although they're not quite of age for us to witness whether or not they have an increased risk of cancer, they do seem to also have structural defects and reproductive tract tissues. And we do know that in mice studies where, because the generation time is shorter, we've been able to, not me personally, but the scientists have been able to take it out to the grandchild generation long enough to see that these grandkid mice also have reproductive tract cancers. So a lot of toxicologists spend time looking at chemicals that mimic estrogens, and we focus on this a lot when we talk about breast cancer risk because of the DES story. And so asking, by analogy, might there be other chemicals out there that contribute in this manner? And so one of the ways that we look at chemicals that could potentially be a risk factor for breast cancer is by simply looking at whether or not the chemicals activate the estrogen receptor. And so on the left is a sample graph where you can see various types of chemicals that have been tested in terms of their ability to activate estrogen receptor. And if you're not familiar with such a graph, the bottom axis means increasing concentration as you go to the right. And if you need a bigger concentration to get a level of activity, then that means it's less potent. So like DES being most to the left is the most potent estrogen amongst these, and DDT is the next most potent. So, I was at the IARC monograph about DDT, and, our final language on that said there was no clear association found between breast cancer and DDT or its metabolite DDE in adulthood, but that they acknowledge that the possible importance of early life exposure remains unresolved. And one of the reasons that is the case is because it's so difficult to get replication in human studies where you have follow-up that takes many generations. So I've been fortunate enough to work with the Child Health and Development Studies, and the woman who is currently in charge of it is the third person who's been in charge of it because of retirement of the scientist leading the study. And she herself is, well, it wouldn't be polite for me to say her name, her age, but she's near retirement age as well. So this was a study that enrolled about 15,000 pregnant women in Kaiser Permanente health system in the 1960s at Oakland Kaiser Hospital. And they were able to measure DDT levels in the pregnant women's blood. And those women went on to have an increased risk of breast cancer. their daughters in their 50s had a higher risk of breast cancer, which we just heard about. And then our most recent study was in the granddaughters who are in their 20s now. Again, that's generally too young to have enough breast cancer cases to do statistics with, but those granddaughters had earlier periods, which we earlier heard was a risk factor for breast cancer. So we're hoping to continue to get study funding to follow them. So I want to touch on a concept called a cancer hotspot, and this is a physical space where a community has more cases of cancer than would be expected normally, just by the rates of cancer historically speaking. There's a couple of breast cancer hotspots that have arisen in the U.S. So in both cases, breast cancer mortality risk was linked to estrogenic exposures. So in Marin County here in California, it seems that higher hormone replacement therapy use in that county was a contributing factor to that excess breast cancer mortality. And remember, that was considered sufficient evidence at IARC. And then that Long Island and New York, higher DDT exposure measured at the time of cancer diagnosis has also been associated with increased breast cancer mortality. So you might hear about DDT and think, oh, this is a chemical of the last century. It got banned in the United States in the 1970s, but it has ongoing relevance here in the US and abroad. This is some research that we put together a few years ago where we measured different levels of environmental pollutants in the blood of people who had immigrated from India to the US on average of 30 years ago. And they had excess levels of a number of pollutants that were banned, as well as some flame retardants. So with respect to the flame retardants, we think that's due to the rigorous fire laws here in California. But you can see that the levels of DDT and its metabolite DDE are much higher in these people. And even though they've been in California on average for literally decades, India still manufactures DDT. So we think that might be part of why their levels are so high. And so for me, this is kind of the new iteration of the silent spraying, because the poison is traveling. So can we identify by activating, if we come back to that concept and visit parabens, which were brought up previously, including by Dr. Boehm, the parabens also can activate estrogen-dependent growth of breast cancer cells and the estrogen. So this is a figure from a British publication. So it's got an O, but this black curve on the far left is estrogen. So you can see the potency of estrogen in causing the growth of these human breast cancer cells. And then, although the parabens of their different varieties, so they're less potent, you need more of them to cause the growth of the human breast cancer cells. These authors put together a very nice visual to show that those levels of parabens that cause that increased growth are the range of concentrations of these parabens that have been measured in human breast tissue. So sometimes when something is less potent, it doesn't make it less relevant. We recently put together a study that methylparaben and propylparaben, which are regarded as safe here by the FDA in the United States, increase the rate of breast, or what's called mammary tumor growth, as well as its metastasis in mice. So you can see that, we're monitoring the mice carefully throughout their lives to check on their tumor growth and make sure they're not sick. And you can see that the total tumor volume is increasing in these gray groups faster than the black dots that represent the control treatment. The levels that we used in this study were at the levels that were considered safe by the FDA. And then at the end of the study, we're able to look at the metastasis to the lung and found that the rate of metastasis to the lung in terms of how much happened during the time period that they were observed was significantly higher for methyl and propylparabens. So in the last minute that I have with you, I wanted to revisit one of the central themes of this great meeting and acknowledge what can we do to reduce the risk of breast cancer. I realize this has got a lot more words than my other slides, but I wanted to make sure to cover everything, and hopefully you can be reading through this as I'm talking. So when we look at the IR carcinogenic agents with sufficient or limited evidence from that table I showed earlier, some of these messages that you should take home is that you can modify your risk of getting breast cancer by drinking less alcohol and smoking or being exposed to less tobacco. With respect to radiation, certainly flying less is a possible way to minimize your exposure. You can also consider contraceptives and menopause therapies that don't contain estrogen and progesterone. That's my timer. I'll just take another few moments. Is with respect to the experimental studies in rodents and human cancer cells, we think that environmental chemicals that activate ER increase the risk of breast cancer. And of course, we need human studies to confirm, but they can be very difficult due to the long time for cancer to rise and numerous difficulties that come about with measuring chemical exposures. So while we wait for that science, please consider the precautionary principle. And as you heard before, read the label with care to look for the opportunity to avoid ingredients like parabens and estrogen-related items, including placental extracts. And with that, I will thank you all very much.
Thank you, Dr. Lamero. And our final speaker on breast cancer risks is Dr. Lindsay Trevino. Dr. Trevino is an assistant professor in the Division of Health Equities and Department of Population Sciences at City of Hope. One of the research projects in her laboratory focuses on the understanding of the molecular mechanisms underlying the link between exposure and parabens, a class of endocrine disrupting chemicals typically found in personal care products and development and progression of breast cancer in Black women. She's particularly interested in the role of EDC-mediated reprogramming in this context. She previously demonstrated that developmental exposure of EDC and by fesol and accelerated epigenetic aging in the development of the liver. This finding highlights the importance of reducing adverse EDC exposures and preventing epigenetic reprogramming and its effect on target tissues. Understanding the molecular mechanisms and underlying epigenetic reprogramming may provide potential prevention strategies for outreach and policy changes and/or therapeutic targets for precision medicine. Dr. Trevino has been selected as a future leader advancing research in endocrinology fellow and Keystone Symposium fellow. She previously served on the Committee on Diversity and Inclusion, on the Governor's Task Force in Research Affairs, Core Committee, and the Board of Directors of the Endocrine Society. Dr. Trevino currently serves on the Science Advisory Panel of Breast Cancer Prevention Partners. Dr. Trevino, thank you so much for your time, and please begin.
Thank you for that introduction. And thank you to the organizers for the invitation to share our work today. I'm really excited about sharing our work. And I also thank Dr. Boehm and Dr. Lamero for really setting the foundation for breast cancer risk. And so I'm not going to go through a lot of that stuff again. That's one of the benefits of going last. As mentioned, in my laboratory, we study the mechanistic link between exposure to endocrine disrupting chemicals, or EDCs, and breast cancer development, particularly through the lens of health disparities. So there will be a separate session on advancing health equity tomorrow, but I want to point out that the studies I will talk about today are related to breast cancer health disparities. And also, as mentioned in the introduction, we are interested in paraben-mediated effects in diverse breast cancer cells. And Dr. Hall, you've heard from Dr. Hall and from others that personal care products contain EDCs, including parabens, which is one class of EDCs that are commonly found in personal care products as preservatives, which we've heard previously. And so our lab's interest in breast cancer was initiated through a collaboration with Dr. Tete and the Bench to Community Initiative, or BCI. BCI is a community-based participatory research project with the goals of, one, developing partnerships with community stakeholders, basic and social behavioral scientists, to better understand the social, cultural, and biological determinants of EDC exposure from personal care products and breast cancer risk. And the second goal was implementing community interventions to reduce breast cancer risk and EDC exposures from personal care products. And so just really briefly, I'd like to pay lip service and just emphasize how BCI was born out of the Cost of Beauty project that Dr. Tete was a part of. And the Cost of Beauty project provided information on the products used by Black stylists in San Bernardino and Riverside counties in California. And in this project, researchers reviewed the products used by the stylists using the Environmental Working Group Skin Deep database. And they developed a list of product usage and categories for a follow-up survey portion of the project. And one thing to note was that parabens were often found in the products they reviewed that were commonly used by the stylists in the area, which was one part of the rationale for focusing on this class of chemicals for our laboratory studies. And as part of the survey portion of the project, which again preceded my time in relation to this project, over 400 men and women were interviewed or collected surveys through the Cost of Beauty project. And the survey results were reported back to the community stakeholders by Dr. Tete. And the common themes that arose from these report outs were, what are these chemicals doing biologically, and how do I reduce my exposure, right? So this is what we've been talking about all day today, and we'll be talking about tomorrow as well. So these became the goals for the Bench to Community Initiative. And there's a lot more history and data that came out of the Cost of Beauty project. So I encourage you to look at the Bench to Community Initiative website or follow BCI on social media to learn more about that if you're interested. So Dr. Tete came to me when I started my independent laboratory and told me about this project and said, what do we know about what these chemicals are doing biologically? So before we started the experiments, we knew, one, that EDCs commonly found in personal care products have been detected in human blood, urine, and breast tissue, right? So we have these measurements. Two, that EDCs alter mammary development, lactation, and susceptibility to tumor development in animal models. We saw that beautifully with Dr. Lamerle's work as an example. And three, that EDCs promote the growth, migration, and invasion of breast cancer cells in vitro as well. So, oh, I don't have slides, so you will not be seeing slides advancing. I just saw a comment. Yes. So this is all well and good that we have this information. That's the basis of what's going on, and it sets a very nice foundation for looking at EDCs and breast cancer risk. However, the studies have mostly been conducted with cell lines and tissue samples from people with European ancestry. So there's little to no data examining mechanisms of EDC action in Black cell lines or samples. So in our lab, we are using Black breast cancer cell lines whose African ancestry has been confirmed. And some of these have been confirmed by Dr. Rick Kittles and his research group. And using cell lines with confirmed African ancestry is crucial for this project. However, there is currently limited availability of cell lines and samples from diverse genetic backgrounds. So I don't have time in this talk, because it's a whole other thing, to go into the myriad of reasons why we have this lack of resources. It includes medical mistrust and a lot of the things, racism in the medical community and in the scientific community. But I will direct you to a recent review we wrote with Dr. Zeynep Madak Erdogan's group in endocrinology that highlights the importance of research inclusivity in this field of research. And given that disparities exist for breast cancer risk and for adverse EDC exposures, it's imperative to develop more diverse models to examine mechanisms of action of EDCs. And this becomes especially important in the context of mixed ancestry, where more and more people are intermarrying between cultural groups and racial and ethnic groups, and highlighting the need to move beyond this idea of self-reported race and ethnicity when studying breast cancer risk outcomes and EDC exposures. And so people might be wondering, but why even study cells in a dish? I mean, how useful is that for cancer research? But actually, in vitro studies have been really crucial in cancer research because experiments using human cell lines have been essential for increasing our understanding of how tissues function, how the disease develops, as well as the effectiveness of drugs and therapies. And so using racially and ethnically diverse cell lines can help identify genes or pathways that may drive the biological contribution to cancer disparities. So, as I mentioned previously, parabens were commonly found in the products used by the stylists that participated in the Cost of Beauty project. We focused on 3 types of parabens in our study: butylparaben, propylparaben, and methylparaben, as these are the most commonly found in personal care products. And according to the Environmental Working Group, products containing butylparaben and propylparaben score high on the hazardous scale, while products containing methylparaben rank moderately. Is it known that there are disparities in paraben exposure? Parabens are commonly found in products specifically marketed to Black women. So there's been a study that has shown that. And studies have also shown that there are racial and ethnic disparities in exposure to parabens, with higher levels of methylparaben and propylparaben measured in Black women compared to white women. And these disparities have also been shown to exist in children as well. So we were talking earlier about how early should we be looking and the exposure timing, right? Although there may be many factors underlying the racial disparity in paraben levels, one potential factor is prolonged use of hair and personal care products that contain high concentrations of these parabens. As Dr. Lamerle mentioned, parabens have been shown to mimic estrogen. Therefore, we initially focused on estrogen receptor or ER positive luminal breast cancer cells. And she also previously mentioned that parabens can promote the survival, migration, and invasion of breast cancer cell lines. But again, most of these studies, or almost all of them, utilized white breast cancer cell lines. So we hypothesize that similar to what has been shown for white breast cancer cell lines, parabens also promote these pro-tumorigenic effects in Black breast cancer cell lines. And this work was recently published in the Journal of the Endocrine Society. If you're interested in specific details, you can look that up. So we treated MCF7 as our white cell line and HCC 1500 as our Black cell line with biologically relevant doses of parabens, and we measured the effects on ER target gene expression and on cell viability. So paraben doses were determined by surveying the literature for studies where these chemicals have been measured in biological samples, including in normal and tumor breast tissue. So we're not looking at levels that no one would ever be exposed to, like really extremely high levels. These are biologically relevant. And what we found was that ER target genes are regulated by parabens in a dose-dependent manner, but not in a linear fashion, which is typical for EDC action. In general, propylparaben and butylparaben were more effective at regulating ER target gene expression compared to methylparaben. And this is consistent with the fact that butyl and propylparaben are considered more estrogenic in general compared to methylparaben. And paraben-mediated effects on ER target gene expression were ER dependent. So when we co-treated with the ER antagonist ICI 180-2780, the effects on the target gene expression were abrogated. We also demonstrated that ER target genes are regulated by propylparaben and butylparaben in a time-dependent manner, with direct ER targets induced at either 6 or 24 hours. Those were the later time points that we looked at. And indirect ER target genes induced at 1 hour. And this is consistent with the pattern seen with estradiol treatment and the respective mechanisms of regulation for either direct or indirect ER target genes. So these parabens are mimicking estradiol in these cell lines. We also observed that parabens regulated ER target gene expression in a cell line specific manner, with the Black luminal breast cancer cell line exhibiting more sensitivity to paraben treatment than the white luminal breast cancer cell line. So that's something that we'd like to look into further with the addition of more cell lines in the future. Since estrogens are known to regulate cell proliferation in ER-positive breast cancer cells, we examined cell viability after paraben treatment, and we found in our hands that treatment with either propylparaben or methylparaben did not increase cell viability in either cell line, but treatment with butylparaben significantly increased cell viability in the Black but not the white cell line. And this effect on cell viability, interestingly, was not ER dependent. So we were not able to block it with co-treatment with ICI 1-AD2780. So more studies are needed to determine the mechanism of paraben action in this context. So with these studies, we have demonstrated pro-tumorigenic effects of parabens in luminal breast cancer cell lines of West African ancestry. And although we focused on cells in vitro, this study exists within the larger context of racist beauty norms and toxic beauty products. It also exists within the scope of structural and institutional inequities, such as residential segregation policies, discrimination, and marginalization that also contribute to breast cancer disparities. So even though this was not the focus of our study, it's important to keep this in mind as we develop interventions to reduce adverse exposure to parabens in personal care products to address known breast cancer disparities in Black women. We've talked a little bit about interventions, but I just want to highlight one way to reduce exposures: to pay attention to the ingredients in your personal care products and replace them with safer product options where possible. And there are apps that can help you do this. One app that I really like is Cleria, C-L-E-R-Y-A. There are other apps, as Dr. Bohan mentioned, but Cleria is really cool because it can be used while you're shopping online at major websites, including Amazon, where you may find some more affordable options for substitution. Because I know that like sometimes some of these substitution products are like really boutique and specialty. But Cleria you can use, on everyday websites like Amazon, and you may be able to find more affordable substitutes. And if you are interested in all natural and clean products from Black owned businesses or brands, you can check out the website Black and Green. It's BLK+GRN, or the Non-Toxic Black Beauty Project from Breast Cancer Prevention Partners Campaign for Safe Cosmetics. So those are some options that you can look into. And then where these studies are going, there's a lot of work to do. This is one of the first studies that has ever looked at diverse breast cancer cell lines. So although we focused on the ER-positive luminal breast cancer cells, we know that Black women are more likely to be diagnosed with triple-negative breast cancer, which is a biologically aggressive form of breast cancer that is harder to treat. And so we've initiated these follow-up studies utilizing triple-negative breast cancer cells to examine the potential role of paraben exposure in this disparity. And in preliminary studies, we have observed increased viability of diverse triple-negative breast cancer cells with paraben treatment. Again, these don't express ER alpha. And we also have focused on breast cancer cells, but what happens in normal cells exposed to parabens? So we've also initiated a pilot study to examine the effects of parabens in diverse human mammary epithelial cell lines to examine the potential role of parabens on cancer initiation. And then I also want to point out that although this session focuses on breast cancer, we also have preliminary data that suggests a pro-tumorigenic role for other cancers that are common in women. In a pilot project, we performed a screen using the NCI-60 human cancer cell panel and various doses of parabens, and that we found that in addition to breast cancer cell lines, parabens increase the viability of ovarian, lung, and colorectal cancer cell lines, as well as melanoma cell lines, which are all common in women. One caveat of this screen is that the cell lines in the NCI-60 panel are mostly, with just a few exceptions, derived from patients with European ancestry, so additional work is needed to define the mechanisms of action in these different cancer types, particularly in the context of disparities. With that, my time is up, and I'd like to thank the people in my lab who contributed to these studies, Dr. Jasmine Tapia, Jillian McDonough, Cesar Gonzalez, and Emily Cobble. And I would also like to thank my collaborator, Dr. Vivek Tete, and the Community Advisory Board members of the Bench to Community Initiative, including Tanya Fairley, Tia Tomlin-Harris, Bing Turner, who also played a major role in the Cost of Beauty project, Maggie Hawkins, and Pastor Rhonda Halbert. Thank you for your attention, and I'm happy to answer any questions during the panel discussion. And thank you for your patience with me not having slides and wondering why they weren't advancing.
Well, thank you, Dr. Trevino, Lamero, and Boehm. We have time for just a few questions before our break. And we'll start with some of those that were in the chat. Dr. Boehm, I noticed that you had answered some of them, but I'll go ahead and go over them briefly. There was one discussant that asked about learning the impact of EDCs on someone that has already been diagnosed with breast cancer. Are there any studies that show the impact on reoccurrences?
One of the things I pointed out was the impact on endocrine disrupting chemicals and treatments. So I've seen in the literature that there is some impact in terms of the endocrine disrupting chemicals decreasing some of the treatments that we use for breast cancer, both chemotherapy and tamoxifen. And I am not sure on the research with women with active cancer and risk of recurrence or survival. I don't have that information. I don't know if some of the other panelists might have that.
And if someone does, feel free to jump in.
Yeah, I think with respect to recurrence, I haven't seen that, but I have seen the evidence on, mortality for breast cancer. So when you have the cluster of increased DDT exposure in Long Island, those women's DDT exposure is associated with increased breast cancer associated mortality, as well as in Marin County, where we had the excess use of hormone replacement therapy and the perimenopausal period that was associated with increased breast cancer there. And then, with the paraben studies, there's been increased metastasis from the low levels of parabens that methyl and propyl in terms of mouse metastasis, and then and the human cell culture. And Professor Trevino, I don't know if you've done metastasis work in your cells at all yet.
We're planning to, yeah.
Good. Well, thank you for that. And following up, are there any reproductive risks, cancer risks, endocrine disruptions, association with generations after reproductive health medications were used by mothers to induce ovulation? And it included Prevent and a couple of the products that are out there, including progesterone, and preemptive studying of these medications on generations following these interventions. Has that been studied? And I think that was for you, Dr. Lamero.
Okay. Apart from, the DES story that I shared with you, I'm not particularly aware of that type of evidence that you're asking about. I mean, one of the things that's really interesting about using medical field science to learn lessons that we can translate into the environmental health is, the doctors keep these great records. So we know the exposures to DES because they're part of the patient records, right? And so one of the ways that we figured out, the general science community, not myself, that the DES was related to increased risk of cancer is because it was associated with this really rare cancer that no one saw in any other context. And so when you're dealing with something like breast cancer, where it's one of the most common cancers afflicting women, it's really hard to track down the exposure because there's multiple causes, right? Whereas in that DES rare vaginal cancer, that rare vaginal cancer is basically only caused by the DES. And so it made the detective work a lot easier, if you will. So, I mean, I think your question's great, but I don't believe that we know anything about that.
Thank you for that. And I'll just finish with one before we transition to a brief break. And this really goes to you, Dr. Trevino, around how do you determine the full genetic makeup of a patient when most persons, particularly in the United States, in the focus study area of Black women, have unreported mixing, meaning they might not appear mixed, but genetic features evidence the mix. And I know this will probably be discussed again in detail tomorrow, but if you don't mind addressing.
No, yeah, that's a great question. And so this is why I made the comment that self-reported race and ethnicity is not, I guess, good enough anymore for like what we do in the laboratory, because you may have mixing and not know, and it really is the genetic ancestry mix and the components, like what came from European, what came from West African, what came from indigenous, right? And we know that risk for breast cancer is different depending on genetic ancestry, and there are groups that are trying to come up with like these risk scores based on genetic ancestry. And then so the way that you do it is you sequence. That's the thing, right? So we need to start thinking about ways to incorporate people of color into these research studies and do this genetic ancestry analysis to be able to have this information. And someone further down the line, if I can just, because it's related, talked about race being a social construct. True. So race is a social construct. It's not a biological construct. That's why self-reported race is not good enough anymore, because researchers in the past have used race and self-reported race as a biological variable. It is not. So when I talk about a race and ethnicity in terms of breast cancer risk, I'm talking about genetic ancestry. So thank you.
And that's a perfect summary of that. Thank you. I appreciate that, because I would have asked the same question. So we have many questions in the chat, but I want to give everyone the chance to have a break. If we can come back at about 5 after. Thank you so much to the three of you for your wonderful presentation. Thank you.
Thank you.
Thank you.
Produced by the U.S. Department of Health and Human Services.